Okay, we are moving on to chapter 27, which is going to focus on prokaryotes and their origins and the diversity of them. It's going to focus on structure and function, origins and evolution, diversity, and ecological significance. Now, prokaryotes are found almost everywhere, wherever there is life, and also places that are too extreme for eukaryotes, like hydrothermal vents and the uh, here in the sulfur mines. So their impact on everything is really huge, especially with humans, especially with ecology. Now, in terms of humans, uh, most bacteria are going to be benign or beneficial in some way to humans, like the bacteria that line your intestinal tract, the bacteria that recycle carbon and nitrogen in the environment, the bacteria that keep fungi from growing in the mouth, the intestinal, and reproductive tracts, and they function mainly as decomposers. Of course, the only ones that you ever hear about are the bad ones, like bubonic plague, which is Yersinia pestis, uh, tuberculosis, cholera, all the STDs. Yeah, you're going to hear more about those than anything else because they're bad, and usually you only hear about bad things. But for the most part, ecologically speaking, and in terms of humans, they're very beneficial or they have no effect whatsoever. Now, um, when I was in school, they had this, I told you about the five kingdom thing, the five kingdom setup with phylogeny, where bacteria and prokaryotes were all crammed into one main kingdom called Monera. And they've since found that bacteria and archaea, which are the two domains that they have now that are both prokaryotic, have diverged very early in the evolution of life to where they deserve to have their own domains. So they went to the three domain system, and then there's domain eukarya, which is also being sort of revamped a little because they're finding out new things about the kingdom Protista in that one. So we'll take a look at it later on in the chapter. The first thing that we're going to go over is structure and function. Okay. Generally speaking, prokaryotes are going to have three main shapes. Okay. You have spherical, which is also called cocci, rod-shaped, which is bacilli, and helical, which is also called spirilla, or spirilla. Most of them will have peptidoglycan, which is a polysaccharide in the chain. It's a polysaccharide chain in their cell wall. The ones that do not have peptidoglycan are going to be all of the prokaryotes that fit into the domain archaea. Now, I'm sure you've heard about this. I'm pretty sure you guys went over this in pre-AP. But they ID whether or not a cell wall has peptidoglycan by something called Gram staining, and it's named after the man who invented it. Where basically you will, if you have a positive reaction, they have peptidoglycan and more simple cell walls. If it's a negative reaction, they have less peptidoglycan and a more complex cell wall, meaning that they have an outer membrane in covering up another membrane. So gram-negative are going to have lipopolysaccharides on the outer membrane, which can be toxic, and these are usually the ones that are going to confer a lot more antibiotic resistance. They're going to have more defense against the immune system. So these are the ones that are normally going to cause all of your illnesses in humans. They also secrete a sticky substance that forms a capsule on the outside of the wall that helps them adhere to cells, kind of like pili would do. Okay, so here's your gram-positive. They stain like a reddish, pinkish color. And then your gram-negatives are going to, your gram-positives are going to stain like a bluish color. And then your gram-negatives are going to stain like a reddish, pinkish color. Okay, you can see here on the gram-positive, they have a simpler wall with all of these polysaccharides, peptidoglycan chain, which is polysaccharides and proteins put together. And then you're also here on the gram-negative, you see that you've got an outer membrane that's composed of lipopolysaccharides, and this is what's going to confer a lot of immunity towards, defense against the immune system and defense against antibiotics, because they have this outer wall here to protect it. Now, let's talk about mobility. Most, let's say, they say half of all, half of all of the prokaryotes are going to be motile, which means that they either have a flagella, which is the most common way here, all right, to move them. They're either scattered all over, the cell, or they're concentrated in one area. They'll have flagella. They'll have a helical filament, which is kind of like a corkscrew. It kind of has this shape to it as well, but it turns in a corkscrew motion, and that's what helps to move it along, kind of like a motorboat would. Or it secretes like a slimy substance that it moves along in, kind of like glides along in, kind of like amoebas do. Now, they are capable of taxis, which means that they can move to or from a stimulus. The stimulus is detected by receptor molecules on the cell surface, so there's all types of taxis. There's chemotaxis, which means that they respond to some kind of chemical stimulus. There's thermotaxis, which is temperature. There's motion. There's kinetotaxis, which is motion. So they're not just very, very simple, okay, I'm going to move here, I'm going to move randomly. They're actually moving to and from some sort of stimulus. Now, if we look at their cellular and genomic organization, they lack membranous organelles and compartmentalization. Right? That's what makes them prokaryotes, is the fact that they don't have a membrane-bound nucleus, and they don't have any specialized compartments in which they do all of their metabolic functions in specialized areas. But that doesn't mean that they don't have specialized internal membranes that are simply folded over. Their genomes are also going to be a lot smaller and simpler in the nucleoid region. They have one double-stranded DNA circle, and then they have a lot of little plasmids which contain a few genes. Usually, remember, if you recall, that they're usually for antibiotic resistance or use of some metabolic nutrient that is unusual for them. But these plasmids replicate independently of the chromosome, and they're transferable. Right? That's what's transferable. And we went over this by transformation, conjugation, and transduction. So I assume that you guys already know how that works. Now, most prokaryotes will reproduce asexually by binary fission in a favorable environment. And what I mean by favorable means that there's enough nutrients, there's enough space, there's, the temperature's right, everything like that. They don't undergo mitosis or meiosis. When a condition gives them an unfavorable environment, they will reproduce sexually, all right? And I say sexually with air quotes because it's not like what we would reproduce sexually with. They do it in 3 ways, all right? Transformation, which is the uptake of DNA from the surrounding environment. Conjugation, the transfer of genes via a sex pili. Or transduction, which is a transfer of genes via the viral mechanism. That's some kind of viral vector. Their major source of genetic variation is going to be mutation, though. And here I'm showing you a picture of an endospore, which is kind of like a hardened, very sturdy cell that can withstand a lot of high heat, high temperature, and things like that in order to kind of wait out the bad environment. So you have this highly resistant endospore. And a lot of these can't even be killed with boiling water. So when the environment becomes favorable, well, then it will shed its outer shell, it will start to rehydrate, and then it will start to grow. And remember that prokaryotes grow very rapidly, they have small generation times, and they are just hard to kill. Okay.